Okay, structure number one, very outside of a plant cell, is the cell wall. Okay, for the function of the cell wall, if they have something about keeping the shape, supporting the shape, okay, supporting the cell, something about support and shape is fine. They have protect, that's wrong. Cell wall does not have any protective function whatsoever. Remember, it's like a house that's been framed with the boards not on the outside yet. Anything can get through it. Okay, serves no protective function. So they got something about supporting cell shape, supporting the cell, something like that. Okay, so that's 1 mark for cell wall and 1 mark for the function. Okay. All right, number 2, the big purple thing, is the nucleus. Okay, and the nucleus essentially holds DNA. That they have something about holding DNA, holding genetic information, okay, something like that. That's good for the function. So storing DNA, yeah. Yeah, I guess that's more the nuclear membrane, but yeah. Okay, for number 3, this yellow stuff with the dots all over it. Rough ER. Okay, I will accept 2 things for rough ER. Transports proteins, makes hormones. Okay, that's what the rough ER does. All right, what's number 4 pointing to then? Smooth ER. Okay, I will accept any one of the following functions for smooth ER. Detoxifies poisons, breaks down complex carbohydrates, makes lipids, repairs membranes, okay, something along those lines, okay. Pardon me? No, doesn't organize anything. Okay, all right, number 5. What's this big thing here? Water vacuole. All right, what does the water vacuole do? Okay, pushes against the cell wall, supports the cell's shape, uses turgor pressure to support the cell. Okay, something about, again, shape and support of the cell. All right, number 6, mitochondria. What does the mitochondria do? Okay, burns sugar for energy would be more specific. Okay. What's number 7? Chloroplast. Okay, and the chloroplast, if they got something about photosynthesis there, good enough. Okay. Okay. For number 7, I will accept microfibers, microfilaments, microtubules, or cytoskeleton, because it's all the same thing to me. Cellulose fibers I will also accept because I had those labeled on the cell diagram yesterday. Okay, what's the function of all of those? Yep, structure, shape, okay, support, something like that, okay. Okay, what's in the middle that kind of fills up all the space? Cytoplasm. Okay, function of cytoplasm, transport. Okay, cellular transport. Stuff can't move through the cell without it. Okay, cellular transport was what we said yesterday. Today we'll be more specific. Today we'll talk about how you couldn't have diffusion. And I think we actually mentioned that yesterday too, diffusion within the cell. It doesn't really hold anything. Stuff can move through it. Well, I just said it filled up the cell. That's not its job isn't to hold stuff up. Stuff moves around. It doesn't have any supportive function. Well, the cell would die without it, but that's true of anything. Right? Any one of those organelles isn't there, the cell's going to die. Okay? The function I gave you yesterday was transport or facilitating diffusion. All right, what's number 10? Cell membrane. Okay, cell membrane. I'm looking for two words here. I told you you had to have these. Listening, please. The two words are endocytosis and exocytosis. If they have those two words, they're good. Okay. Number 11 is the Golgi apparatus. Processes waste, packages waste, exports waste, something like that. Waste, yeah. Okay. You have something about moving waste out of the cell for the Golgi. And number 12, the purple dot inside the purple thing. Nucleolus. I will accept two functions for the nucleolus. Copy DNA and facilitate or control protein synthesis. Okay, those are the two functions of the nucleolus. Pardon me? It doesn't really sort it. No, the DNA is kind of everywhere in there. Okay? Sorts is not correct. Okay, listening here. It's out of 24. There's two for each one for the name, one for the function. Give them a mark out of 24, put it on the desk right here. All right, this spot in your notes, we're picking up where we left off from yesterday. We're talking about the methods that a cell will use, guys, the methods that a cell will use to transport materials. Okay, we talked a little bit about diffusion yesterday, but we'll finish that one up. Diffusion is the most important. Okay, it is the most important method of intracellular transport. Okay, what does intracellular mean? Inside the cell. Okay, everything gets transported inside the cell through the cytoplasm using diffusion. Yes, the rough ER can do some transporting, the smooth ER can do some transporting, etc. But in the end, anything moving through the inside of the cell, the cytoplasm, has to move by diffusion. That means it moves from an area of high concentration to an area of low concentration, which usually gets it where it needs to go, because it's usually in low concentration where it needs to go. Okay? But this is not a targeted transport method. Okay? This isn't Purolator or Canada Post. It isn't addressed to a certain place. It is not going to a certain place. It is simply going from high to low, which means it's going to end up probably where it's needed, but probably other places as well. It's really kind of a shotgun approach, okay, to transporting materials. So the further away the destination is from the source, the less concentrated that material will be when it gets there, right? That's kind of the drawback of diffusion. It works fine as long as it doesn't have to go very far, which is why cells are typically pretty small. Okay? It works fine over cellular distances. 100 to 200, maybe even upwards of 300 micrometers, it's going to work pretty reliably. Anything above that, and diffusion is really starting to break down and not be effective. All right? Okay, and remember also that it's passive and thus requires no energy from the cell, which is why cells use it. Okay, it just occurs naturally. Okay, our next one is osmosis. Osmosis is only going to transport one material. Okay, osmosis will only move water. Okay, it'll only move water. So it's key that we know that. Osmosis is only going to move water. And it doesn't move it within the cell, it only gets it into the cell. Okay, so it is moving it across the membrane. Okay, so osmosis is the movement of water across the membrane. It is also passive. The cell has no control over this process. Okay, which means most of the time, osmosis works just fine. But under strange circumstances, osmosis can cause a cell to die. Okay, those strange circumstances would be extremes in concentrations of salts. Okay, water moves across membranes in response to concentrations of salt. It always moves towards high concentrations of salt because that's where there is less water. Okay, so it's really moving down the gradient, sort of, okay, for water. Okay, where there's lots of salt, there needs to be more water. Water moves that way, right? So if I've got, let's say, a cell, okay, that has that is kind of a normal cell, but outside of it, there's really, really high concentrations of salt. The water will move out of the cell and into the salty water, and the cell will shrivel up and die. Okay? This is why you can't drink salt water. Okay? The same process happens in all of your cells. So if you are, let's say, in a lifeboat in the middle of the ocean, right, and you're looking around going, water, water everywhere, okay, not a drop to drink, you're absolutely right. Don't drink it. Because if you do, you'll die of thirst quicker than if you don't drink anything. Right? Because as soon as you consume that salt water, the amount of salt in your body goes up. Okay? And what happens is your blood gets saltier. And naturally, water begins to move out of your cells and into your blood in order to make the concentration in your blood and in your cells equal. It's always seeking a balance. Any transport that is passive is seeking a balance. So water begins to move out of your cells and into your blood. And then your body detects suddenly that, wow, the blood is like full of water. We got to get rid of some water here. We're going to have high blood pressure. Okay? So it signals to the kidneys to start pulling water out of your blood. Now that gets rid of a lot of the salts you've consumed, but it also gets rid of all the water you've consumed. Okay? And so as a result, it's not just getting rid of the salt water you drank, it's getting rid of probably about another 50 to 75% of the water in your body in order to do that. So for every liter of salt water you drink, you'll probably lose between 1.5 liters and 1.75 liters worth of your own water to get rid of it. So you dehydrate really quickly if you are drinking salt water. Okay? This is also why if you've ever eaten like really salty food, after you eat it, you're thirsty. Okay? It's because your body's starting to draw water out of your cells in order to kind of get rid of all the salt that you've consumed, so you get thirsty. Okay? Everybody follow me there? Okay, so that's how osmosis works. It's going to have water move from low salt to high salt across the membrane. So those are the key points. Across the membrane, okay, to balance salt. Right? It's water only. No, osmosis can happen pretty quickly. Yeah, it's a process that happens fairly quickly. Now, the other condition that can happen is if I put a cell in completely distilled water that has no salt in it, water rushes into the cell because the cell has salt in it. And what's going to happen? It's going to break. The cell will pop. Okay? Because it can never achieve a balance. There's no salt outside, and even a small amount inside, you can never balance that out. There's never going to be any salt outside, so the water just keeps going in, going in, going in, until pretty soon the cell just swells up and goes and pops. Okay? And then that's going to be it for that cell. Okay? So osmosis, in extreme cases, can cause either the dehydration of a cell, okay, the shrinking and then kind of death of a cell, or a rupture. Okay? If you put salt on a slug, that's osmosis. Okay? It doesn't burn the slug. What it does is slugs don't have skin like us. Their skin is much more membranous and mucousy. Okay? When you put salt on them, water just rushes out of their tissue to balance the salt. Okay? It tries to get kind of more of a slimy layer on them so that the salt kind of just washes off, but it just sucks the water out of them pretty much right away. Okay? And they just kind of shrivel up. Okay? It's because of osmosis. All right? Is that making some sense? Okay. All right. So, big things about osmosis. Like we said, passive transport of water, it's across a membrane, okay, and it's trying to gain equilibrium. Okay? It's trying to get a balance of salt concentrations on either side. It's not the movement of salt, it's the movement of water in response to salt. Okay? So cells use this all the time. It's how you maintain kind of a fluid balance within your body. But it's also important for plants. It's how plants transport water. When water evaporates from the leaves of a plant, the salts that were in the water when they absorbed it out of the ground are left behind. It's like if you boil away water, all that crusty stuff gets left behind inside the pan. Okay? So all the salts get left behind. Well, that creates a very salty condition in the leaves, and water moves naturally up the tree in order to balance it. And it's that process that pulls the water along with the polar nature of water, okay? So the cohesion and adhesion up the tree. Okay? It's osmosis that provides that pulling force. So it can have a pretty big effect even on a large scale, okay? On a cellular scale as well as a large scale. Okay? Also, when I was talking to you yesterday about what happens when a plant starts to wilt, okay? When a plant starts to get dehydrated, okay? So I know a plant cell is not supposed to be perfectly round, but let's just say it is, okay? If I start to get a lot of salts building up because water is being drawn out of the cell, the concentration of salts inside the cell goes up, and water from the vacuole starts to move out because, again, of osmosis. A cell cannot control this process. It just happens. Okay? A cell can't go, oh, this is bad, stop osmosis right now. It can't. Okay? Osmosis just happens. So water eventually starts to get drawn out of these vacuoles, and then the cell can't support itself, and the plant wilts. Okay? That's what causes that process as well. Okay. So what I would do, guys, on a test, okay, on a unit exam or something like that, is I would give you a situation. I would describe, let's say, I've put a cell in salt water. Describe what process is going to occur and why. Okay, so as soon as you see water, okay, you're probably not thinking diffusion anymore, because what's the only process that transports just water? Osmosis. Okay, so you got to identify what process, what are the key characteristics of each process. Well, diffusion is within the cell only, okay, and it can transport anything within the cell, okay, and it always moves from high to low, and it's passive. Osmosis is across the membrane. It's water only, and it's passive. Know the kind of key identifiers of each one. Okay, and the last one is the complete opposite of the first two. Active transport. All right, not passive, active. The cell uses energy to do this. Okay? If a cell is going to use energy to get something inside, it must need it pretty badly. All right, so active transport is just that. It is active. It's pumping solutes, and it's pumping them against the gradient. Okay, it's not just allowing it to go there from high to low. It's going from low to high. Okay? And it's using energy to do that. Just like water doesn't flow uphill naturally. You need to pump it uphill or carry it uphill. You have to expend energy to make it go that way. Right? So active transport is actively moving stuff from outside the cell, which you can see in the diagram here, into the cell. Okay? In order to do that, it's got to use carrier, or we usually say transport proteins that are on the membrane. Okay? These proteins change shape when energy is applied to them. Okay? So normally they're in this open shape where they're open to the outside, and then when they're activated, they close like this, and whatever was in the channel at that time gets forced into the cell. So they just kind of open back and forth, okay, like this, and that requires energy, but it actively pumps stuff into the cell. Okay, so active transport, like we said, pumping solutes against their gradient. Okay, it uses energy. Okay, it's also across the membrane. Okay, and yeah, that's kind of the big things here. Okay, so expends energy, actively pumps substances across the membrane. And again, active transport can transport anything. Right? Osmosis is the only transport process that only transports one thing. Okay, diffusion and active transport can transport anything, but active transport uses energy to do it. Okay. Making sense there? Those are the big three. I ask about all of them on your unit exam pretty much every year. Okay? I'll have a question that asks about each of them, so you've got to be able to identify them and explain to me how they work. Okay. Now, other ways that cells can move things. These are not as important because they don't happen as often. Okay? Exocytosis is movement of things out of the cell. And we already said the Golgi apparatus is pretty much responsible for getting things to the membrane, and then the membrane takes care of it from there. Right? So exocytosis involves a vesicle, okay, a bubble, that has stuff in it, whatever it is, hormones or waste products or whatever, okay, that are going to be exported from the cell. Right? So this vesicle, which is made of the same stuff as the membrane, heads towards the membrane. And when it gets there, it merges with the membrane, and when it does that, essentially it opens it up, and whatever was inside goes out into the intracellular fluid, okay, which is basically the fluid that surrounds all of your cells. That usually has quick access to the blood, okay, and the blood can transport materials quickly to the rest of the body. All right. Everybody follow me there? So exocytosis is, okay, when we got stuff, okay, vesicles produced by the Golgi apparatus, okay, is moved to the cell membrane, okay, and then pops out into the surrounding fluid. Okay. Usually things that are moved by exocytosis are also very large. Okay. So large that they would not be able to get through the membrane on their own. Okay. The membrane, remember, is made of all those fats, all those lipids. Okay. And these molecules simply can't get through that layer. Okay. So they have to be kind of escorted out in this vesicle. All right. Transport into the cell is endocytosis. Okay? There's 3 ways a cell will get things that are too big to go through the membrane by active transport or osmosis. Okay? And osmosis obviously only moves water. Okay? So if things can't get in by active transport and they're too big to go through the membrane by diffusion, then they have to be engulfed. Okay? And the process is the same, just sometimes it's a solid, sometimes it's a liquid, or sometimes it's a hormone or a special molecule that the cell needs to take in quickly. Okay, so there's 3 types of endocytosis. There's phagocytosis, which is the engulfing of food and solid particles. Pinocytosis, which mechanically is the same. The membrane just kind of pinches in a little bit and fluid gets trapped. Right? So if osmosis isn't moving stuff quickly enough, the cell will just gulp water using the same process, using pinocytosis. Okay, so cell drinking. Okay, and then lastly, receptor-mediated endocytosis, which has to do with hormones. Okay, so you can see here that we got phagocytosis first, okay, and that's where we're engulfing a solid particle. Pinocytosis here, and receptor-mediated endocytosis. You see, all those things that I showed you were on the cell membrane the other day, those are involved now, the receptors. Okay? When a hormone signals a receptor that it is present, the cell membrane automatically engulfs it. Okay? It's just the receptor tells it to do it immediately. Food particles, sometimes they have to bump into the cell several times before the cell goes, oh, hey, right, that stuff's food. Engulf that. Okay? But receptors for hormones and things like that, that's an immediate process. Right? Hormones have to be taken into cells to affect the cell processes immediately. All right, so like we said, with phagocytosis, the cell engulfs the particle by wrapping a pseudopod. Okay, that's essentially, but pseudopod means false feet. Okay, so it's like engulfing it like this and sealing it in. So I think I used the analogy of picking up after your dog the other day, right? You wrap your plastic bag around your hand, pick up the poop, and then turn it inside out. That's what phagocytosis and pinocytosis is like. If you could like grab a handful of water by that same process and turn it inside out, that would be pinocytosis. Okay? All right, so this is how your white blood cells kill bacteria. Okay? They essentially eat them. All right, pinocytosis, mechanically, the same as phagocytosis, except it's not engulfing a solid, it's engulfing fluid, okay, in order to get a fluid balance reestablished quickly. Under an electron microscope, it looks like this. You can actually see the membrane folding in and trapping a whole bunch of fluid in each one of those things, okay. What they do is they essentially dehydrate a bunch of cells and then throw them in distilled water and they just start gulping like crazy. Okay? Osmosis happens really fast and they start gulping and then they just explode. Okay? Like very shortly thereafter. But that's how they get a picture like that. Normally a cell would not be engaging in pinocytosis to that level. Okay? It would maybe have 1 or 2 as opposed to like 8. All right, and then in receptor-mediated endocytosis, okay, similar idea here, and this is like again an electron microscope picture of this process, but you can see it almost looks furry along here, okay, and that furriness is caused by the receptors. This is about the only receptor you can see with any reasonable ability there, okay? It's kind of this Y shape, and then you can see the hormone molecule right here that's attached to it, okay, and that's signaling the cell to take it in. That's happening all along this little arch here. Okay, it's going to take all of that in. You can see it once it's engulfed it. You can actually see the heads of all those receptors with the molecules attached to them. Okay, being engulfed and taken into the cell. Right, so that only happens. Okay, with essentially hormones and other molecules. Okay, so usually proteins and other kind of large molecules will use receptor-mediated endocytosis. All right, questions on those? Okay. All right, I want you guys to answer those questions, and we'll go through them here in a few minutes. Okay. Color blindness is caused by a person's cells being unable to produce certain proteins. Okay, where does the problem lie? It's in their DNA, okay? Not in their ribosomes, okay? If there was a problem with their ribosomes, they wouldn't be able to produce any proteins. Okay, they would die, yeah, okay? Not having ribosomes is kind of fatal, okay? Yeah. So being colorblind simply means that there's a mistake in your DNA, and as a result, the protein that is produced does not work right. We talked about that yesterday. The example we used yesterday was color blindness and diabetes, okay? Not being able to produce insulin properly, okay? All right, for number 2, why is it important for organelles to have their own membranes? Not really. Think about what goes on in a couple of different organelles. Inside the nucleus, all your DNA is stored. Okay, kind of important stuff. In the lysosome, chemical reactions that involve the digestion of cellular material occur. Okay? In the mitochondria, reactions that involve burning sugar for energy occur. Do you want all those taking place in the same area? Absolutely not. Having its own membrane involves, or it allows it to have the right conditions for each process. That makes your cell more efficient at each process. Okay? That's that compartmentalization. The importance of compartmentalization means all the cellular processes occur at a much greater level of efficiency, and all of the cellular structures are essentially protected from each other. Okay, if the lysosome's membrane ruptures, what gets out? The digestive enzymes. And what will they do to the cell? Eat it. Yeah, okay? That's how cells, okay, essentially kill themselves if there's something wrong with them. Okay? That's their kill switch. If they are malfunctioning and they recognize it, they will rupture their lysosomes on purpose, okay, in order to destroy themselves and keep them from becoming cancerous. Okay? Number 4, what problems might a cell have if all the ER was removed? Unlike the one-word answer I just heard, okay, what are some of the things a cell would no longer be able to do? It would not be able to detoxify poisons, produce hormones. Well, the ribosomes make those. Transport proteins we got already. What about the membranes? What will it not be able to do to them? It won't be able to repair them. No lipids. Can't make lipids if you don't have any smooth ER. Okay? Won't be able to break down complex carbohydrates for fuel, okay, into glucose, which is what your mitochondria need. Right? So basically, a lot of things would go wrong. Okay? The last result, of course, which would be death. Okay? But we want to have more than that, because if you wrote that for a 5-mark question, I would give you 1. Okay? All right. Questions on any of those? All right.